and of course the idea that I had and other people had was that, well, if you can take a gene and move it around, maybe you can use it to treat a disease. And so I went and spoke to one of the first people who was wanted to do that. He hadn't done it yet. His name is French Anderson, and I worked in his lab for a year and asked him, how can I become a gene therapist? And he said, oh, it's simple. Go to medical school and learn about the diseases and then go back to the lab and start working on them. Yeah, right. Really simple. I had a PhD, and I decided, like, I had a job offer, a postdoc offer, but I decided to apply to one medical school, and I got in, and I went there, and I actually did what he recommended. I went to medical school, learned about the diseases, did a little bit of training afterwards, and then went back to the lab.
Thank you, Jean. Thank you, Jean, for coming into
Sure,
this
sure,
session.
sure. No, it's a pleasure to meet all of you, and what a diverse group. It's really great to hear about your experiences. So, so I had a very circuitous career. I started out in zoology as well as one of you, and cell and molecular biology. But basically, molecular biology hadn't been developed when I started, and so I learned it as it was developed, and started working first on in marine invertebrates, but then I went higher up in the evolutionary scale and started working on mouse development. And at that point in time, people were making the first transgenics, and there was I got involved in that field, and it was so excited at the thought that you could actually take a gene and put it into a new, in this case, a fertilized mouse egg, and you get the mouse to grow bigger. I'm referring to the growth hormone studies that happened early and around, that must have been about 1981 or so. And I went, and of course the idea that I had and other people had was that, well, if you can take a gene and move it around, maybe you can use it to treat a disease. And so I went and spoke to one of the first people who was wanted to do that. He hadn't done it yet. His name is French Anderson, and I worked in his lab for a year and asked him, how can I become a gene therapist? And he said, oh, it's simple. Go to medical school and learn about the diseases and then go back to the lab and start working on them. Yeah, right. Really simple. I had a PhD, and I decided, like, I had a job offer, a postdoc offer, but I decided to apply to one medical school, and I got in, and I went there, and I actually did what he recommended. I went to medical school, learned about the diseases, did a little bit of training afterwards, and then went back to the lab and started working on gene therapy. But this was before we had any vectors. We knew anything about the diseases in the eye. I started working on diseases which would have been way too difficult for gene therapy because they were multigenic. But then I had a collaboration with somebody who was working on the eye, and I realized, wow, the retina is a perfect target because of the immune situation, the separation from the blood supply, the fact there are two of them, that they're not, the diseases aren't usually life-threatening, et cetera. A lot of different advantages. Plus, there are surgical techniques, and you can see it. You can dilate the pupil and see the back of the retina. And so I started working on that, with physicochemical reagents to introduce genes to the retina. And then when I came to Penn, shortly after I came to Penn, Jim Wilson arrived at Penn to set up what was called the Institute of Human Gene Therapy, and brought a vector core which was making adenoviruses. And I learned how to make those viruses there, and then started testing those in the mouse eye. And lo and behold, they did transfer a reporter gene. In this case, it was LAX-Z at that time. into the retina really efficiently, but the expression he kind of waned after about 2 weeks and disappeared. Um, and I used that as a first proof of concept in a really quick, fast-moving retinal degeneration mouse model. It's a mouse model of retinitis pigmentosa. So, and it worked, but of course it was short-lived because of the clearance of the transgene and probably the virus. And then as vectors were developed, I started testing those in the retina. And there were a bunch of vectors I tested, but adeno-associated virus really worked the best. And we moved quickly, going from using at this time GFP as a reporter gene. We tested that in a number of different species, not just mouse, but also dog and monkey. And lo and behold, it led to long-term transgene expression. Our monkey study went 2 years, and the expression that we could visualize using GFP persisted pretty much the same intensity over that period of time without causing immune infiltrate or anything, and no serious like problems with the eyes there. And so we started working on, like what could be a good target to show that this is effective for gene therapy. And I had started working on dogs with adenovirus, which for the same reasons it didn't work very well in the mouse. It didn't work well in dogs, but using the adenovirus, we had developed the surgical techniques to deliver genes to dogs and knew what to look for in terms of safety and how to analyze the tissue and so forth. And then lo and behold, there were 3 little puppies born which had RPE65 deficiency. And so we generated a vector which contained the RPE65 and delivered it to one eye of each of these dogs. And these dogs were blind. They were fearful. You'd go near them to pet them, and they'd jump because they couldn't see your hand coming towards them. And they'd crouch in the corner because they were scared, of whatever was going on in the facility. But within 2 weeks, they started running around playing and watching the caretakers, the people who were bringing the food and cleaning the cages and so forth. And I got a call. I wasn't expecting anything to happen really quickly, if anything at all. I got a call about 10 days after we did the injection saying, Gene, I think your dogs can see. So of course I rushed over to the facility, which is about an hour away by car. And yes, the dogs could see. It was really pretty obvious. So we did some crude testing just to show that they could play with each other, that they could catch a ball, that they could run around and not bump into things, and realized pretty soon that this really impacted their ability to see in dim light. That even in the well-lit animal rooms, the affected animals couldn't see, but after the treatment, they could run around and not bump into objects and so forth. And so we did a lot of testing in these animals using the conventional tests, which are electroretinograms and looking at pupillary light reflexes and other tests. And I also measured their ability to walk around a room that was full of obstacles and avoid those obstacles. And it was pretty dramatic. Two of the animals we euthanized looked at the tissue, and it was great. One of them we maintained. He became a very famous dog. His name was Lancelot. And he lived till he was 10 years old, which is about average for these large dogs. But during that time, he was brought to the US Congress several times to be an example of the importance of biomedical research. And he didn't embarrass us. He didn't bite anybody. He could see things that people were throwing to him and stuff like that. And he also served as the father of numerous other puppies with his lovely wife, who was affected, his wife and sister, who was affected with the disease as well. So they served as the breeders for a line of dogs that we then used, affected dogs that we used for additional experiments, looking at different AAV serotypes and different transgene cassettes and looking at immune responses and all sorts of things, and looking at durability of expression. And so that data became the basis of moving forward to a human clinical trial, where we tested this cautiously, first in adults, just to make who were severely affected. And they showed benefit. And then we moved to children as well as adults. And the vast majority of the individuals showed improvement. And one of the biggest outcome measures for improvement was ability to see in dim light. Like the dogs, they could see in the dark and walk around and not bump into things. And that matched up with some of the clinical tests of light sensitivity. But we moved forward with that and then tested the ability to go to the other eye. If you inject one eye, is it going to be safe to inject the other after antibodies had developed to the AAV, which they did. Humoral antibodies did develop to AAV. And it was safe to inject the other eye. The first eye didn't lose any benefit. The second eye gained benefit. And with that data, we move forward to what's called a registrational clinical trial, a phase 3 clinical trial, aiming to get approval of this as a drug. And that started in around 2012, 2013, and involved 2 sites, our site at Children's Hospital of Philadelphia and a second site in Iowa. And it enrolled 29 individuals, children and adults, and the vast majority of them showed a really remarkable benefit. There were really minimal adverse events, no really serious immunologic issues. There was one person who had, in the early phase trials, who had endophthalmitis, which is a bacterial infection, but that had nothing to do with the AAV itself. But we've been following the individuals now in long-term follow-up. Some of them are hitting the 15-year mark. This has been a lifetime, like a career-long experiment. But over the course of that time, I've had the opportunity to get to know these people really well. The children are now grown up. They're having children of their own. They've gone to college. They have jobs, getting married. And I've been to some of the weddings and visit their families. So I feel incredibly lucky to have been able to observe not only the clinical trial, but to see how these people are doing in the real world. And so one of the big questions is, how long will this last? Well, I don't know. I'm beginning to see signs that it's waning. It's still way above baseline, but there's a whole long process where the company needs to go through data locks and making sure the data is like analyzed in a certain way and so forth. So my analyses don't, they're just kind of, I'm anticipating what's going on, but the company has the final say about what the data says. But the bottom line is, it's been safe, it's been long-lived, it's changed the lives of these people, and set a path for development of gene therapy, I hope. now what is going on, and so far there's not been a second approved retinal gene therapy, and that would be the biggest boost, I think, for the field. There have been a number of trials which have gone on through phase 3, but they failed to meet their endpoints. Three of them, two of them in particular, run by Biogen, and I think that's because they picked the wrong endpoint. So that's one important lesson, that it really, you can't just speed through this. You have to pick the right endpoint, and like to be sure you're going to like hit your mark. But the bottom line is it's appeared to be really safe with AAV. There are occasional reports of inflammation, particularly with some of the high dose vectors. There's been inflammation if the material's been injected intravitreally and there's no steroid coverage. But subretinally, it's been pretty safe. And there was one clinical trial for Leber's hereditary optic neuropathy, which is a mitochondrial disorder, and another one for retinoschisis, where there is a lot of inflammation that and they treated with steroids. The Leber's hereditary optic neuropathy trial showed some benefit, but unfortunately it showed benefit in both eyes, not just one eye. And so it was not approved. But subsequent to then, there's some evidence of cytoplasmic transfer, which might explain why the second eye also showed benefit. In my view, it's like, wow, you're getting two eyes improved. That's great. But the regulatory authorities didn't like that. So the bottom line is, what are the inflammatory episodes which have caused concern, and I associate those with potential immune response. So things that maybe your expertise could help with. Most of the things which clinicians have deemed immune response have to do with what they see on imaging. It's not a biopsy, so we don't really know what it is, but the main imaging of the retina is what's done with what's called optical coherence tomography, or OCT. And that creates essentially a cross section of the retina, like what you would see if it was a piece of tissue sliced and looked at under a microscope. And in some instances, they've seen little bulges in the layers, which they interpret to be immune infiltrate. Whether they are for sure, I don't know, but it makes people scared. And so they treat these retinas with steroids. And a lot of these little deposits will disappear, which suggests that they are indeed immune infiltrates. And certainly in the animal experiments that are done, for example, with monkeys, with high doses before the humans are injected, just to look at the worst possible effects, you can see those kind of, in histologic sections, you can see that there are little infiltrates in the outer retina and in the retinal pigment epithelium and choroid, which probably correspond to what people have reported in the humans who've had these consequences. But steroids have mostly kept those things under control. I suspect that one of the reasons why people get these infiltrates is because there's a lot of empty vector in the preps. So there are a lot of differences in the vector preparations. They're made by different groups, made using different methods and purification steps. And people don't always report how pure the vector is. In other words, they may be giving a dose of 1E11 vector genomes, and that would be full vector genomes. But if there are a lot of empty capsids in there, there could be 10 to the 14th vectors in there, and a lot of them are just empties that aren't counted. And so I think that's a really important area to look at in terms of immune response, what could be causing that. let's see, what else? I've heard of two individuals who had a pretty severe immune response after Luxturna was delivered to them. And unfortunately, this was during the pandemic lockdown, and the PI of that study offered to send me PBMCs, which I don't know what I would have done with them if they had arrived, but unfortunately they arrived like 5 days after they were drawn and they were dead essentially, because of the trouble getting shipments and everything else and receipt of shipments during the pandemic lockdown. So I don't really know what happened, but that there's certainly a need for people to follow up on things like that. And now I've been involved with a few other clinical trials since the Luxturna, one a choroideremia clinical trial, where again we use very pure vector. No steroids were used at all in these patients. They all did really well. There were 15 of them. And I'm currently involved in a study on another form of congenital blindness, which is going really well. There have only been 3 patients injected so far, but so far no concerns. So I think vector purity is a really, like a big issue, but let's see what else.
Yeah, that's
Yeah,
good, Gene. Maybe some of the questions that these we folks have may uncover something. So just raise your hand if you have any question, then you can ask. I mean, Bhavya, I know you had some when you. Yeah, why don't you go ahead?
Hi, thanks for all that talk. It does. I know you weren't prepared, but it did sound very related to what we're trying to do. Well, uh, one of my questions when you said there was a different kind of immune response to how the injection was delivered, so intravitreal versus subretinal, and how do you decide on how that is done or which is the best place to deliver?
sure. So in our experience, our laboratory experience, the only way to target cells in the outer retina, such as photoreceptors and retinal pigment epithelial cells, the way to target those efficiently is by subretinal injection, because that puts the material in direct apposition to those cells. So it's taken up really, really efficiently. And there's been work by other groups to try to develop vectors which could be injected intravitreally and then migrate through the retina to target those places. But in my experience, I'm skeptical of how efficient those things are. But the second part of that is that we saw that if we inject AAV into the vitreous, the center cavity of the eye, it can target the ganglion cells, but it's also more likely to cause inflammation. And that was mainly through large animal studies. You can inject a lot of things in a mouse and not see any problem, but in a dog, you can see inflammation. And let's see. So, and we initially realized the differences in those two sites when we're using adenovirus, which is really inflammatory, much more inflammatory than AAV. And there's a dramatic difference between what happens if you inject intravitreally versus subretinally, with the subretinal being much less likely to cause inflammation. Now, of course, there can be leaks when you inject subretinally, leaks from the subretinal space into the vitreous. And so that could account for some of the inflammation that people have seen in different studies. So the surgical approach can be physically damaging and can cause some issues.
Yeah.
But
I think Shweta has a question, and
yeah.
Shweta has been working on infectious diseases in the eye. I don't know if the question is related to that.
Uh, Not really related, but Dr. Jean was mentioning that about the outcomes in clinical trials. And I just wanted to understand what would be a good set of outcomes that one would need to look at so that you avoid some sense the failures in clinical trials. If one is looking at a new trial for a new intervention or a therapy, what would be a good point to start?
You're talking about safety outcomes or?
Safety. So that also. So you did mention there have been many other trials, but not all. Many of them have failed, right? So
Yeah.
what kind of outcomes would how would you redo that study so that you get clearer, more
Okay.
positive data?
Well, one of the challenges is that there's traditionally only been one outcome measure that's been used to approve new drugs, and that's reading the eye chart, seeing the E, the A, different lines on the eye chart. And the rule is that if you show an improvement of 15 letters on the eye chart, that's 3 lines on the eye chart, that's significant. There's a lot of variability that we all have day to day and like how many letters we can read, but 3 lines is a pretty robust metric. The problem is that and reading, of course, is an important thing to all of us. It's a clinically meaningful outcome because you can do more things in your life. But the problem is for a lot of these diseases, most your visual acuity, your fine visual acuity is accounted for by an area that's about 10 nanometers in diameter. It's the fovea. It's your central vision, which is right in the back of your macula. And if the gene therapy reagent does not target that region, there's no way it's going to improve. um, and in a lot of these diseases, the fovea is already degenerated. You're not going to restore it. And so you can't count on visual acuity as being improved. And in some diseases, visual acuity is pretty good until late stage in the disease. For example, in choroideremia, which is one of the trials that failed, people have 20/20 vision, even though if they drop their pencil, they can't find it because they can't see anything beyond this tiny little central tube. And so I think it was the wrong outcome measure to choose for those trials. It should have been an outcome measure that measured function of the more peripheral retina, which would have been something like light sensitivity or visual fields or those kind of outcome measures.
Yeah.
So those were high-profile failures. They involved a lot of money in the trial, a lot of people participating in the trial, and they failed, even though people thought that they were getting better.
Can I just, add to this? I think, well, because I was working with the group which was doing, it's called Best One. Now it's with Opus. But I think that was also an example of picking the wrong endpoint, and the exact same thing. Visual acuity is what the FDA wants, but in a research setup, there is a test called FST. I know you've also spoken about it in the past. And now, so something new, a new test. How are you convincing the FDA to say, okay, these are my new endpoints, but never heard of them, but this is new,
Right.
and this is what you should be
Right.
measuring because that's important.
Yeah. So, and that's a subject that's highly discussed with retinal specialists now. So to get Luxturna approved, we developed a new test, a test where the person has to walk through an obstacle course under different light conditions and they're graded on how accurately and quickly they move. And this was actually based on what I observed in the dogs early on, that the dogs could walk around and not bump into things after treatment under dimmer light conditions. And so the test is called the MLMT, the multi-luminance mobility test, the one in humans. To get that approved, we ran a validation study in affected individuals and individuals who have normal vision, and showed that it reliably could identify level of visual function, or functional vision, we call it, in these patients, that it was really a quality of life measure. So that's the important thing for the FDA, that it's a quality of life measure. So we tried to get them to approve the FST, which is a test with a machine where people push a button if they see light of a certain, and the light is shown at different intensities and wavelengths. And that's a robust test, but the FDA said that does not show clinically meaningful result, because people don't care what it looks like if they push a button. But if they can walk around and not bump into things, that's meaningful. So that's why the FST is not approved as a test, even though it's a fantastic test and it correlates with the results of these other tests.
See. Well, just to add on it, you did mention immune infiltrates and looking at OCT. Well, I'm working on adaptive optics and, its ability to measure at cellular level. So do you think that has potential to, just have meaningful endpoints in research?
Yeah, well, I think it's meaningful to the clinicians, but again, the FDA would say, who cares if there's a little blob there if it doesn't affect what the person can do day to day?
I
So
see.
you'd have to relate it to some other metric, like if you illuminated a little spot using your adaptive optics and the patient could see it after treatment, but not before treatment or something.
Yeah.
Has to be functional.
Yeah.
Got it.
You had raised your hand. Did you have a question or? Yeah, Bhargavi, go ahead.
Ma'am, as you have mentioned earlier about the empty viral capsids, like where a gene of interest is not inserted into that viral vector. So in that case, when the dosage is given to a person, and if most of the viral vectors are empty, you are saying that there are high chances of immune responses. So in that cases also, the efficiency of the treatment, does it goes down because the viral vectors are empty? Also, second thing, in that case, if the efficiency is down, has the treatment to be given again in those cases?
Nobody has gone back to an eye that's directly to an eye that's been injected and re-injected it for fear of immune response. We did a study in monkeys using Luxturna where we showed we could do that safely. There's some physical damage, like from the first injections. Most of the damage is from the surgery itself, but there was no inflammation due to a second administration of vector. But that has not been attempted in humans. I don't know who will be brave enough to do it the first time.
Yeah. So,
Ma'am, one more doubt I have. So, in this treatment has like particular criteria, like before the patient has been selected for a treatment, like we need to have two faulty genes. So, in that case, in spite of the gene, do we have to look at the damage level also, like the density of healthy cells in the patient, so that the treatment is effective?
Yes. Well, I think the inclusion or exclusion criteria will describe, you don't want to inject this into somebody who's totally healthy because there's risk with any procedure. So you definitely have to have pretty good information indicating that in the case of RP65, that both of the alleles are faulty. And there have been instances where we question whether one of them was faulty, and did cell culture studies and so forth to try to figure that out. It's not always really clear. I'm sure some are worse than others. I don't know what I haven't gone over the data to look to see if there's better responses in people who have 2 null mutations, for example, versus for some small expression, etc. But that would be interesting to know.
Yeah.
Thank you, ma'am.
Praveen, you have something quick to ask?
Yeah. Actually, I enjoyed reading more about your journey and all, and I read that you were inspired by Professor Anderson, and the journey how you started into the cell and gene therapy. So my question is like how you met Dr. Catherine Hai, and how the Spark Therapeutics came into this picture, and what was this transition from the academics to the commercialization? How if you could help us to like this story, I'm more curious to know about it.
Sure. Well, I first met Kathy Hai at a retreat of the Institute of Human Gene Therapy. It's that institute I told you that Jim Wilson started in about 1994. It was a retreat in the Poconos, which are small mountains an hour or two away from Philadelphia. We shared a room together. And lo and behold, we were both interested in gene therapy. And we also had 3 children that had almost exactly the same names: Sarah, Catherine, and she had a John, but I had a William. But they were about the same ages, and the older 2 went to the same school. So it started out kind of on a like fellow faculty with similar family structure thing. And I'd run into Kathy at various meetings, and she and followed her work with a company called Avogen in the Bay Area of California. That she brought me out to that company to try to get people interested in doing the RP65 gene therapy, and they weren't interested in it. But then that company folded, and then there was a big disaster in the gene therapy field where Jesse Gelsinger died, and that was in an adenovirus trial. But Kathy had the wisdom to ask Children's Hospital of Philadelphia to support gene therapy, to hire people who were being laid off and bring in the expertise. And she persuaded them to give her money to run 2 different clinical trials. She was going to do one on hemophilia because she's a hematologist. And she knew about my data. So she knocked on my office door and said, "Jean, how would you like to run a clinical trial for RP65 deficiency?" And I'm like, "What?" Of course, the answer was yes. And so that's when we started working together. And I can tell you the exact day that happened. It was July 21st, 2005. Because it's burned in my memory.
Yeah.
Wow, that's a great story. Yeah.
I really enjoyed this story, and I also like found that you struggled initial days in getting funding for for their studies and all, and later like once the data has been done, like all these companies come together, and even you know like the Novartis came to get that license, and after that Roche acquired the Spark. So this was very like the commercial part, how things goes in terms of money and all. So
That's amazing.
Yeah, well, you were asking about how Spark was formed. And so when we had our first set of data from phase 1, we shopped it around to a lot of big companies, and a lot of them were really interested, and they'd come and look at it. But we got really nervous that if a company took it, it would stop right there. that they did not have the motivation or the expertise to push it forward. And we realized if this is going to get to the finish line, we have to do it. So that's why we set up the company. And that I can remember that day too. That was in Boston, sitting outside at a cafe. It was like, okay, we're going to start a company. And it was, and that company was funded by Children's Hospital largely, like half of the company. It started small, and did not attract the support of those big companies until much later.
Thank you. We're on the top of the hour, and it's almost 10:45, 10:40 here in India. So thank you again, Jean. This has been amazing. It's always great to talk to you. I just saw those pictures recently where Vibha and you went to the Indian temple. I was telling everybody about them.
That was amazing. Yeah, it was so fun.
Yeah.
So do you all know Vibha?
Yeah, no, some of them know. Yeah, some of them know Vibha, right?
Okay.
Yeah.
She's a dear friend. It's wonderful for me now to know her two children who are
Yes.
student at Penn.
Yeah, no, I'll be back. I'm usually in Philly from April to October, so I'll come and look you up when I'm there. So
That's you choose a good time to stay away. It's so cold now.
it's so cool now. Yeah. So thank you again, Jean. This has been a pleasure. And
You're welcome. It's great to meet all of you.